Hello, everyone, and welcome to today's live broadcast, Signaling Pathways at Your Fingertips, Multiplex IP to Targeted Mass Spectrometry Assay Kits for AKT mTOR Pathway. It is presented by Bhavin Patel, MD, a Senior R&D Scientist for Protein and Cell Analysis at Thermo Fisher Scientific. I am Judy O'Rourke of LabRoots, and I will be your moderator for today's event. We are delighted to bring you this educational web seminar presented by LabRoots and sponsored by Thermo Fisher Scientific. For more information about our sponsor, visit www.thermofisher.com. Before we begin, I would like to remind everyone this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on the Ask a Question box located on the far left of your screen and type your questions into the drop-down box that appears on the screen. We'll answer as many questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, please use the Ask a Question box to let us know that you're having a problem. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located in the top right corner of the presentation window and follow the process to obtain your credits. Please join me in welcoming Dr. Patel. I will now turn the presentation over to him.
Thank you for the introduction. So today we'll talk about the signaling pathways at your fingertips, Multiplex IP to Targeted Mass Spectrometry Assay Kits for AKT mTOR signaling pathway. So just to kind of give you an overview, I'll start out with background information on targeted proteomics and mass spectrometry, and also talk about the advantages and challenges for targeted mass spec assays. And then moving on to how do we optimize target enrichment method as well as verify the different reagents to develop this targeted mass spec assay, and also how we can put together in a commercially available kits for this multiplex IPM assay. And then I will kind of talk more about, in the last part of the presentation, how do you get the absolute quantitation of these AKT mTOR pathway targets using these multiplex IPM assay kits. We'll compare the Western blot method with this new IPMS assays workflow, as well as how do we develop these AKT mTOR pathway assay kits, and also talk about the application. So the first question we ask, like, what does mass spectrometry, protein mass spectrometry offer to biologists? So currently, Western blot is the gold standard method for study of the signaling pathway proteins. As we know, these pathways have multiple proteins involved in them. And at the same time, we ask questions that, a lot of times we are targeting these pathway proteins. So we must be looking at different drug combinations with different concentrations. So that generates a lot of different conditions. So with the Western blot, you can only do single target at a time. And if you are comparing with multiple conditions, it's a lot of long process of generating a lot of Western blot, where that's what biologists are kind of, looking at the protein mass spectrometry, and what it offers is detection of protein isoforms and modifications that cannot be seen by other methods, as well as identify and quantify multiple proteins at the same time, identify their post-translation modifications such as phosphorylation, as well as the protein-protein interactions. And mass spectrometry can be combined with other antibody-dependent methods to improve the specificity, such as like immunoprecipitation or flow cytometry can be used upstream of the mass spectrometry. So what are the challenges in a mass spec-based proteomics? Even though mass spec provides power of identification and quantitation, however, it will not identify all the proteins present in a given sample. So the challenge here is because of the wide dynamic range of abundance of this protein. As an example, plasma has 10 to 12 orders of magnitude dynamic range in terms of different proteins present in a sample. And also, even though there has been a significant amount of progress made in the last decade for mass spec instrumentation sensitivity, but there are still limitations in terms of what you can see in a mass spec. There is also another effect we call ionization suppression, where the abundant species will mask the identification of the low abundant species due to the ionization suppression effect. And when you are doing this with this mass spectrometry, a lot of times you get missing information for quantitation. So it is very important to design the experiment with the solutions outlined here, where reducing the sample complexity makes it more palatable and easier to quantify. Using target enrichment strategies allow us to carry biological context through our workflow. And also, second most important thing is the most accurate quantitation by mass spectrometry requires the addition of stable isotope label internal standard, which can be used to generate standard curves of known concentration ranges. Also, it allows you to compare multiple samples relatively within the same LC-MS-MS run. So knowing this challenging in mind, how do you reduce the sample complexity? As I mentioned, most commonly used biological samples such as biofluids, cells, and tissue have a 10 to 12 orders of magnitude dynamic range of relative protein abundance, making it nearly impossible to identify low abundant proteins over the abundant ones. And of course, we make it even more difficult by using the proteolytic enzyme to digest these thousands of proteins into millions of peptides. That's why a lot of times the depletion, enrichment, and fractionation methods are used before or after digestion steps in order to detect these low abundant proteins as well as their post-translational modified forms. And I'll talk more about optimization of this enrichment step by immunoprecipitation method before mass spec application. So how do we get target-specific protein extractions and enrichment? We have optimized this IP to mass spec protocol and have developed the mass spec compatible IP kits that includes the protein AG or streptavidin magnetic beads. It comes with all the reagents necessary to perform the IP, which includes cell lysis buffer, optimized binding method, mass spec compatible wash buffers, and elution buffer. And also this eluate is compatible with the direct in-solution digestion method downstream. So if we kind of walk through, on the left-hand side is the typical immunoenrichment method that we propose, where you start with the lysing the cells, and then you take the lysate and antibody and allow the antigen-antibody complex form overnight. Then day 2, you bring in the magnetic beads to capture this antigen-antibody complex. You perform the washes, and wash buffers are mass spec compatible. And then you use the mass spec compatible elution buffer to elute the antigen of the beads. Once you get the eluate, you spiroact dry, you reduce alkylate and digest to generate the pool of peptides. And these peptides are further separated by liquid chromatography by using their, based on the hydrophobicity factor. And as each peptide elute off the column, they will get further fragmented, and that generates the fragment spectra for each peptide. And this spectra is matched against the theoretical spectra to identify the peptide sequences. So on the right-hand side in a table, multiple targets from AKT-mTOR pathway were screened across 2 cancer cell lines, with and without enrichment. And as shown in a enriched IP column, we were able to get more unique peptides identified after IP compared to niche samples. So hence, enrichment enables the better detections by mass spectrometry. So in the second part of the talk, I'll talk more about how do we develop these multiplex IPMS assays. And I'll start out with optimization of target enrichment protocol and verification of these targeted mass spec assays, and then development of the commercially available kits. So the workflow for verification of antibodies by IPMS and development of targeted MS assay. In step 1, we select the targets of interest. We select the relevant cell models and antibodies for antibody verification by IPMS. And in order to, we typically use the messenger RNA and deep proteome data generated from this cancer cell line to identify the abundance level of specific targets in relevant cell lines to screen in a step 2. In a step 2, we perform immunocapture and LC-MS analysis, where we select multiple antibodies for a given target to perform the IP, and followed by LC-MS analysis to verify antibodies capture efficiency as well as specificity for each antibody. And we try to select the antibody with the best capture efficiency to further bioisolate and use them in a multiplex IP. And in the third step, since we are generating this directed discovery data when we verify antibody by IPMS, this data can be used to identify the chondrotypic peptides that can be used with the targeted PRM SRM assay using the aqua peptide. So I will talk a little bit more about the targeted mass spec methods that are commonly used. So there are two methods that are commonly used currently in the field. One is selected reaction monitoring, also known as SRM. Those are typically done with the triple quadruple mass spectrometer, and that's the one is shown on the top panel. And in the bottom panel is the second method, which is known as parallel reaction monitoring, PRM, and those are done with the Thermo Scientific Q-Exactive hybrid quadruple mass spectrometer. So how do these two methods work, and what are the common things and differences between them? So SRM method, the Q1 and Q3, the first and last mass analyzer of a triple quadruple mass spectrometer are used as a mass filters to isolate a peptide ion and a corresponding fragment ion, and those are the ones that are shown in a light green color. And the signal of the fragment ion is then monitored over the chromatographic elution time. And the specific pair of M over Z value associated to the precursor and fragment ion selected are referred to as transitions. So once you get the transition optimized, you can select multiple transitions for a specific peptide, and you can monitor them for quantitation. In a PRM method, in the bottom panel, again using the three quadrupoles, the third quadruple is replaced with the high resolution and accurate mass Orbitrap mass analyzer, which allows us to monitor all fragment ions that can be detected for each peptide. The advantage of monitoring all fragment ions is ability to identify and exclude interfering fragment ions from the unknown samples during the data analysis steps, and that allows us to get much more accurate quantitations for each peptide. So how do we develop these targeted mass spec assays using the aqua peptides so that we enable the absolute quantitation? Here are the specifications for aqua ultimate grade peptides that we use. They come with the plus or minus 5% concentration precision, more than 97% peptide purity, as well as more than 99% isotopic enrichment. And aqua peptides are used to generate the standard curve spanning from 200 femtomoles as the highest and 0.08 femtomoles as the lowest by serial diluting the aqua light peptide and keeping the constant amount of aqua heavy peptide. And that gives you light over heavy ratio, and you plot that against the concentrations that was used for light peptide serial dilution. And as an example here, the AKT1 peptide 7-point standard curve shows that we get a really good linearity and at least 2 to 3 orders of magnitude dynamic range. And on the right-hand side in the graph, we have selected target peptides from 12 AKT mTOR pathway targets, and that allowed us to get linear quantitations and 2 to 3 orders of magnitude dynamic range by both PRM as well as the SRM methods. And as you can see here, we were able to get lower limit of quantitations up to 0.08 or 0.23 femtomoles that covers all the target peptides. And essentially, PRM and SRM method provided the same level of sensitivity. So once we have validated the antibody mixtures as well as the peptides, this is kind of outlining the development of the Thermo Scientific SureQuant multiplex IP to targeted MS kits. These kits are developed for simultaneous enrichment and quantitation of multiple total and phosphorylated proteins from the AKT mTOR signaling pathway, As you can see on the right-hand side, there are two tables. The one panel is for AKT total targets, and the other panel is the phosphorylated forms of the same AKT mTOR pathway targets. Now, these multiplex panels comes with the absolute and relative quantitation module. So each panel has a total or phosphorylated has IPM Spec sample prep module that you can do 10 different reactions using the bioaccumulated antibody mix, positive control lysate, step-targeted magnetic beads, wash buffer, trypsin, as well as other reagents are necessary to do the mass spec sample prep. There are two different quantitation modules available that goes with the IPMS sample prep module. If you want to do the absolute quantitation, you can use absolute quantitation module that comes with the aqua heavy peptide mixture, aqua light peptide mixture, as well as 6 protein digest as a matrix, and system suitability standard to assess the LC-MS-MS system performance and dynamic range. On a relative quantitation module, since you are not generating a standard curve, it only comes with the aqua heavy peptide mixture and system suitability standard. Both these quantitation modules comes with the validated instrument methods as well as the skyline analysis template. So in the next part of the talk, I'll discuss more about how do we get the absolute quantitation of AKT mTOR pathway targets using these assay kits. And we'll start with going over the Western blot workflow. That's the current gold standard method, and then comparing this method, Western blot, with this targeted mass spec assays, as well as the key characteristics of these assays and some of the supporting data with that, and then also talk about the applications of evaluating these two methods and ask questions like, can we get absolute quantitation of AKT mTOR pathway targets, and how the quantitation differs between the two methods. So to start our workflow for Western blotting, the Western blotting workflow, also known as the Western workflow, is comprised of 3 major steps. You separate, transfer, and detect. The purpose of the Western workflow is to provide researchers with a technique to separate proteins from biological samples by molecular weight, and then visualize their protein of interest using a variety of detection methods. So here you can see on the bottom panel, there are some representative Novex and Pierce products for each step of this procedure. By integrating the product portfolio from Novex and Pierce, Thermo Fisher Scientific is now able to provide the most comprehensive end-to-end set of products and instruments for the Western workflow. Now, in contrast to Western workflow, this is the workflow for multiplex IPMS assays. We have selected a total 12 total and 12 phosphorylated protein targets from AKT mTOR pathway, as well as 12 total targets from RAS, MAPK pathways for this multiplex IPMS assay development. We have validated antibody for each one of these targets and used those and bioaccumulated them and created IPMS module that I previously talked about, as well as the peptide for quantitations. So in a second part of the workflow, that you utilize all these validated reagents to perform the multiplex protein immunocapture. And you can also automate this process on a Kingfisher instrumentation. And then after you have this mass spec samples prepared for mass spec analysis, you can perform the LC-MS analysis with either directed discovery or using the targeted PRM, SRM method using the aqua peptides. And in the end, targeted mass spec data analysis was performed using the Skyline software to quantify absolute quantitation of each target in a single mass spec run. So how do Western blot and this IPMS assays compares with each other? Western blot has been a gold standard method for protein quantitations. And the advantage of doing the Western blot is the low cost, the very easily available antibody, because there are a lot of antibodies available that are Western blot validated. It's relatively standardized workflow, and it requires very less amount of samples to do the Western blot. Some of the challenges with this method is the specificity is lower. You do not get absolute quantitation. It's a single plex, so you have to do single target at a time. It's not very feasible to investigate the entire pathway in a very short period of time. So that actually results in a low throughput. If we look at the multiplex IP to targeted mass spec assays, the advantage is allows you, since you are performing the immunoenrichment step, allows you to get a better detection and quantitation of low abundance targets with very high specificity, accuracy, and reproducibility. Also allows you to get the absolute quantitation by running the standard curves. Since it has a multiplexing capability, it also improves the throughput. And with this method, allows you to study the protein-protein interactions as well as the post-translational modification analysis. And you can not only investigate the entire pathway, but you can also investigate the adjacent pathway with this method. And I'll talk a little bit more about how we study the adjacent pathways using this method. Some of the challenges with this new technology is there are not that many validated antibodies available in the market that are verified by IP to mass spec. Also, it comes with a relatively high cost. The sample prep method is a little bit relatively complex, and also since the cost, main cost associated with the LC and MS instrumentations, you do need to have access to have LC and mass spec instrumentations with this technique. So far we talked about validating the antibodies and validating the peptides. How can we put all these things together for enrichment of AKT mTOR as well as the RAS or pathway targets? So on the left-hand side, it shows the representation of the RAS pathway in orange and the AKT mTOR pathway in a purple color. And here, these are the most two important signaling pathways involved in a cancer biology. And the targets that we selected are highlighted with the red asterisk that we developed the targeted mass spec assays for. On the right-hand side, the table shows the mass spec data for total and phosphorylated pathway targets. AKT mTOR RAS pathway proteins were immunoprecipitated from human IGF-1 stimulated HCT116 lysate using the Thermo Scientific Pierce MS-compatible magnetic IP kits. These are streptavidin magnetic beads before the mass spec analysis. And as you can see from the data, we were able to detect protein isoforms as well as interacting partners for AKT, mTOR, RAS, and PI3K targets. Not only you detect the isoforms and identity of the target, we were able to identify the relevant phosphorylation site that contains the phosphopeptides, but also detected for AKT1, AKT2, mTOR, RAS40, PIK3R2, RAF1, and RASK1 during the mass spec analysis. And mTOR and interacting proteins from mTOR2 complex were also verified with this approach. So once we have developed these assays, this is kind of giving you a little bit more in-depth analysis of how the data looks like by doing the directed discovery mass spec analysis. So the panel A and B are multiplex IPM assay for total and phosphorylated AKT mTOR pathway targets, and panel C is the multiplex IPMS for total RAS or PIV pathway targets. And our specifications to get the relative quantitation is identifying at least 2 unique peptides for each target, and also get more than 1 e to the 6 peptide area for each one of the unique peptides that were identified for each target. And using this multiplex IPM assay kits, 12 total and 12 phosphorylated AKT mTOR pathway proteins, as well as 12 total RAS or pathway proteins were enriched from IGF-1 stimulated HCT116, a cell line using the bioactivated antibody mixtures and the streptavidin magnetic bead-based mass spec compatible magnetic IP kits. And nano-LC-MS analysis for each assay resulted in identification of all these targets, which meets our relative quantitation specifications. And also for each target, we were able to get less than 20% CV when we calculate the top 3 peptide area, and those are the ones that are shown as a error bar on each bar graph. As you can see there, we were able to get a really good percent CV for each one of these targets, even with the directed discovery DDA kind of mass spec analysis. And then we also developed this PRM quantitation for each one of these target peptides. So here on the top panel, multiple peptides for 12 AKT mTOR pathway proteins on the top panel, and the bottom panel is the RAS pathway target peptides. We were able to monitor them with the linear quantitation R-square value of more than 0.996 and more than 3 orders of magnitude by doing the PRM mass spec targeted analysis. And PRM quantitation limits of the peptides for AKT mTOR as well as the RAS pathway targets are shown here as on a bar graph. The upper limit of quantitation for each peptide was at 500 femtomoles on column. And as you can see, we were able to detect a lot of these targets with a lower limit as low as 0.05 femtomoles on a column. So how does this overall assay works when we combine the multiplex IP as well as the targeted MS quantitation? So this is kind of showing you the robustness of the AKT pathway phospho multiplex IPMS kit, where we had 3 different operators, and each operator was performing the IP in triplicates from start to finish. And we were able to, our specification for PRM quantitation is individual CV should be less than 15%, and the combined CVs are less than 20%. So if you look at here, for each target peptide we were monitoring, we were able to get the CVs, we were able to meet the specs for the percent CV, as well as most of the target peptide we were able to monitor them with the less than 10% CV. And if you look at the robustness of the AKT pathway total multiplex IPMS kit, as you can see here, again, we were able to, with the three operators, each operator in a triplicate, we were able to get the CVs less than 15% for almost all the peptides. So overall, both of these AKT mTOR total and phosphorylated multiplex panels, we were able to get the CVs as low, like in a single digit for a lot of these target peptides. So once we have this AKT mTOR and the RAS assays, we wanted to ask a biological question where we say we took 2 cancer cell line, HCT116, which is a colorectal cancer cell line, and A549, which is a non-small cell lung cancer cell line. And we stimulated both these cell lines with the human IGF-1 growth factor, which stimulates the AKT mTOR as well as the RAS pathway. And we inhibited this pathway using the PI3K inhibitor LY294002. And since PI3K inhibitor is actually kind of common between both the pathway, we wanted to see how the protein expression changes downstream as well as upstream of this PI3K inhibitor for AKT mTOR pathway targets as well as the RAS pathway targets. And as you see here on both signaling pathways, the targets that are highlighted with the red box are the ones that we were monitoring to assess the protein expressions with the different conditions. So as I mentioned, we have developed these multiplex IPM assays for AKT, mTOR, as well as the RAS pathway targets. And on the top panel, what you're seeing is the Western blot results, where in HCT116 cell line, the 4 different phosphorylated targets, the PRAS40, phospho-AKT, phospho-P70S6K, and phospho-mTOR, from Western blot, you can see that PRAS40, AKT, and P70S6K phosphorylated forms were upregulated after stimulation with the human IGF-1, and they are also downregulated after inhibition with the drug followed by stimulation with human IGF-1, whereas we were not seeing significant changes in a phospho-mTOR upon the drug inhibition. And if you look at the bottom panel, that's the representation of the multiplex IPMS assays for AKT mTOR pathway targets. The blue bar is the untreated, the gray one is the stimulated, and the blue one is the inhibition followed by stimulation from the same HCT116 cancer cell line. As you can see that we are stimulating a lot of these pathway targets upon the human IGF-1 stimulation, such as PRAS40, AKT, P70S6K, mTOR, phospho-IRS1, GSK3 alpha, and beta. So all these targets were upregulated upon stimulation. 4 out of all these targets were downregulated upon the inhibition. So phospho-AKT1, phospho-P70S6K, phospho-mTOR, phospho-IRS1, they were downregulated upon PI3K inhibition, where there were not significant change found in phospho-GSK3 alpha and beta, as well as the PRAS40. So we do see some correlations between these 2 techniques, Western blot as well as the multiplex IPMS assays. There is the variability between these 2 techniques for some targets, specifically phospho-AKT and phospho-mTOR, and that could be due to the antibody specificity because you are using different antibodies for different methods. One thing I do want to point out here, when you look at the Western blot for phospho-related AKT forms, since we know that AKT has 3 isoforms, AKT1, AKT2, and AKT3, you are measuring the total amount of all 3 isoforms on a Western blot. When we do the mass spectrometry, you can actually distinguish AKT1 from AKT2 and AKT3. And here we can see that AKT1 is being downregulated upon the drug inhibition. So just to kind of summarize this 2 cancer cell line with the stimulation and inhibition study, this is how the protein expression changes for the AKT mTOR pathway targets and the RAS ERK pathway targets in a 2 cancer cell line with and without drug treatment. So we didn't see that many changes in the total AKT mTOR pathway targets in both cell lines, regardless when you stimulate or inhibit and then stimulate. Multiple phosphorylated AKT mTOR pathway proteins showed upregulation upon human IGF-1 stimulation and downregulation in a PI3K inhibited HCT116 cancer cell lines, except the GSK3 alpha and beta. In A549, on the other side, the phospho-AKT mTOR pathway targets were upregulated except mTOR, but they were not inhibited upon the drug inhibition, except the phospho-IRS1. When we look at the total RAS and ERK pathway protein showed increase in a protein expression in both cancer cell line, and in a phosphorylated forms on the RAS and ERK pathway targets, we didn't see any change in HCT116 cell line, but on the other side, we are seeing downregulation in A549 after the drug inhibition. So we are seeing very different protein expression changes for both of these pathway and the two cancer cell lines. So we are seeing cell line dependent differences in a response to the PI3K inhibition. So just kind of summarizing the talk, enrichment by immunoprecipitation is necessary for identification and mass spec quantitation for these low abundance signaling pathway proteins, also as well as to study the protein-protein interactions and post-translational modifications such as phosphorylation. And the short quant AKT pathway multiplex IPMS kits, by using that, we get a higher yield of pathway proteins and very few non-specific bindings that allows you to get a better signal to noise and better quantitation for these pathway targets. Also, this total and phosphorylated targeted multiplex IPMS kits allow the simultaneous quantitation of multiple AKT mTOR and RAS ERK pathway proteins, as well as the modifications in a treated cell line. Now, elucidating the specific pathway differences between A549 and SCT116 cells will lead us to better understand and treatment of the lung and colorectal cancers. And the last benchmarking study of multiplex IPMS assays against the Western blot using the super signal West Dura substrate and in Vitrogen iBright FL1000 imaging system, overall we do find a good correlations between for both AKT mTOR and the RAS pathway proteins. However, there are some variability between techniques for some targets, and that could be due to the different antibodies are used for different methods. And then you also have the specificity are different for capturing these targets with the different methods. So that's all I have, and I would be happy to take any questions that you have. Thank you so much for your time.
Thank you, Dr. Patel, for that informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question drop-down box located on the far left of your presentation window. Type your question into the box that appears on your screen and click the Send button. We'll answer as many of your questions as we have time for. Let's get started. Our first question is, do you use phosphopeptides for the phosphor assay kit and non-modified peptide for the total assay kits?
That's a really good question. So we have spent a lot of time understanding the quantitation of these phosphorylated forms of targets using the phosphopeptide. And the challenge we see is a lot of times the phosphopeptides, analytically, they are not really good for quantitation. So to answer the questions, what we do is we use the antibody that are specific for the phosphorylated form of target, but after the enrichment and digestion, we spiked in with the non-modified version of the unique peptides for each one of these targets. So we don't use the phosphopeptide for quantitation. We use the native form of unmodified peptides that we know has a really good dynamic range, has a really good lower limit of quantitations, and gives you much highly accurate and precise quantitation for both total and phospho kits. So the both total assay and the phospho assay are utilizing the same unique peptide, and we rely on different antibody to capture total and the phosphorylated forms at the protein level.
Thank you. Do the mass spec assay kits work with tissue and mouse xenograft samples?
Yes. So I haven't shown any data on those, but we have used this same assays on a normal as well as the tumor tissue lysate as well as the mouse xenograft samples, and we were able to identify a lot of these targets from the cancer or tumor tissue lysate as well as most of the targets in mouse xenograft samples.
Does it require a different lysate preparation?
Yes. So the cell lysate is pretty much straightforward cell lysis. With the tissue samples, you do have to you can still use the same lysis buffer, but you have to perform the rinsing the tissue and then homogenization, followed by lysis using the same IP lysis buffer, and it works with that method.
Great. How does this protein-level immunoenrichment method compare to the peptide-level enrichment?
So we are performing at the protein-level immunoenrichment by validating the antibody by IPMS method. You can also generate the antibody for very unique peptide for each one of these targets and take the lysate, digest, and do the peptide level enrichment. The advantage of doing protein level enrichment is not only you can enrich your target, but you can also get the protein-protein interactions as well as the isoforms information, and also you can get the stoichiometry information about the unmodified and modified forms of at the protein level.
Great, thank you. Looks like we have time for one more question. It's a two-parter. So the first part is, how do you verify the antibody selectivity and capture efficiency by IPMS?
So I kind of went very quickly on a workflow, but typically we start with, we have done extensive deep proteome analysis for 12 cancer cell lines. So we kind of know the relative abundance of a lot of these important signaling pathway targets in each one of the cell lines. And we start with selecting multiple antibodies that are commercially available for specific targets, and we screen all of them from the control cell line that we know there is a relatively medium abundance of that particular target by using the mass spec compatible IP kits that utilizing the protein AG magnetic beads. And once you run all these samples side by side, when you analyze the mass spec data, what we do is we calculate the fold enrichment of the intended target by comparing with the control IP, and higher the fold enrichment we get, we tends to select the antibody that gives you the higher fold enrichment. And also we have this, there is another continuation from there, that we also have developed a new method where we not only look at the enrichment of the target, but as well as the epitope we can map the epitope binding site for each one of these antibodies that we have validated. So we can confirm that this antibody is indeed binding to the epitope that is designed for, and it's very valuable for the phospho-specific antibody.
Thank you, Dr. Patel. Here's the last part. How do you select peptides to use for each target quantitation?
So once we verify this antibody by IPMS method, we use the same data to ask questions. So what are the unique peptides do we see in a mass spec? And we tend to use that data and analyze them and ask questions, which peptides has a very good response factor in a mass spec, as well as reproducible and stable. And then we screen multiple peptides to assess their linearity, stability, response factors. We order them in a aqua format, and then select at least 2 target peptides for each one of these targets in our final assay.
I would like to once again thank Dr. Patel for his presentation. I would also like to thank Thermo Fisher Scientific for making today's educational webcast possible. Before we go, I want to let everyone know that today's webcast will be available for on-demand viewing through September of 2019. You will receive an email from LabRoots letting you know when this webcast will be available for replay. Please share that announcement with your colleagues who may have missed today's live event. That's all for now. Thank you for joining us. We hope to see you again soon. Goodbye.